Well, this is our second session on gametogenesis, and in this session, we will learn about the formation of the female gamete, which is called the egg. And this process of the formation of female gamete in the ovaries of mammals is known as oogenesis, which means the birth, genesis of the egg, oogenesis. Now, there are various stages that are involved in this particular process, and this process takes place in the ovary. Now, there are a couple of things that we need to know about the ovary, and the first thing that we are going to look at is the broader structure of the ovary. Now, the ovary is divided into two parts: the outer region, where we see these large structures which are known as follicular structures, and this region here, the outer region, is known as the cortex, cortex, cortex. And then we have the central region of the ovary, the region where you see these blood vessels. This region is known as the medulla. Now, cortex is the region where all the processes that are related to oogenesis take place. So the processes that are related to the formation of egg or the female gamete take place here in the cortex, and the medulla is the region which supplies the ovary with nutrients via blood, and also takes away the waste materials out of the ovary that are generated as a result of metabolic activities. And let us start. Where does this process of oogenesis takes place? Now, oogenesis starts with something known as a primordial germ cell. And where are these cells? Now, here we have the ovary, and we have the outermost layer. And if you look at this outermost layer of cells, I hope you know that the outermost layer of any particular organ is made up of a tissue known as the epithelial tissue. And here, the outermost layer of the ovary not only protects the ovary and gives it a structure, but also is generative in origin, which means it gives rise to more cells. So there are these germ cells in this epithelium. Germ cells means the cells that give rise to more cells. So these particular cells of the germinal epithelium are known as primordial germ cells, and these primordial germ cells undergo mitotic division. Mitotic division is conservational division, by which we mean that if a cell undergoes mitosis, its ploidy will be retained. Let us assume that this is a haploid cell. If this undergoes mitosis, it will form two daughter cells which will be haploid. If this cell, the parent cell, happens to be a diploid cell, then undergoing mitosis will produce two daughter cells, which both of them will be diploid. So this is mitosis. Just revising a bit of it with you, though in order to be a part of these lectures you have to be thorough with meiosis and mitosis. Anyway, so primordial germ cell undergoes mitotic divisions, and it produces large number of cells. Some of those cells, some of those primordial germ cells will replace the primordial germ cells. See, the germinal epithelium is going to lose cells, so it undergoes mitotic division to add up cells to its own self, which are lost as a result of wear and tear, the regular wear and tear. But there are certain cells which do not add up to the primordial germ layer, but then decide further to undergo meiotic events, and these cells are called oogonia. These are cells equivalent to spermatogonia that you studied in your spermatogenesis. Now, these oogonia then prepare themselves for meiotic divisions. But how? Now, a cell that has to divide, whether it has to undergo mitotic division or whether it has to undergo meiotic division, which is the reductional division, a cell, let us assume a diploid cell, or we will only consider diploid cell. A cell has half the DNA content from the parent and half the DNA content from the father, half from the mother. So parents contribute equal amounts of DNA. So before a cell must divide, it doubles up the content of the DNA. So what happens before mitotic event, during the interphase period, the cell actually doubles up its DNA. So let us say this is a cell, it doubles up its DNA, and then this oogonium actually becomes something known as a primary oocyte. Now, oogonium forms primary oocyte by multiple mitotic divisions. The characteristic of oocytes is that oocytes have double the amount. They are all diploid cells. All of them are diploid. Have a look at this. This is a diploid oocyte, but the primary oocyte has twice the amount of DNA compared to the oogonium. So I hope this difference is amply clear to you. Now, the primary oocyte. Now, where is this primary oocyte? This primary oocyte has been formed in the ovary of the female during the embryo stage. Now, remember that oocytes are formed even before the birth. So, the process of oogenesis starts even before the girl child is born, unlike in males where gametogenesis starts only after puberty has been reached. So now we've got primary oocytes and they are diploid. Now in this particular ovary we do not see any primary oocytes. That is, it is because the primary oocytes are located in special structures, in particular structures which are known as follicles. Let me move the slide around, and I've magnified this ovary. Now these primordial follicles, say let us use a marker. The germinal epithelium gave rise to oogonia, which gave rise to oocytes. The primary oocytes are generated by this primary germinal epithelium, and those primary oocytes are then here, and a layer of cells grow around them. You see, so this central cell that you see here is actually the primary oocyte, and this is the primary oocyte is still. Let us go back and let us take a look at the image. The primary oocyte is diploid, but its ploidy, but its DNA content is doubled up, so it is still diploid. And here is the primary oocyte. Now the primary oocyte is surrounded by cells. It's something like this. So let's say this is the primary oocytes. A layer of cells grow around this primary oocyte, and these cells are called granulosa cells. Granulosa cells, and this entire structure is called a follicle. These granulosa cells are also called follicular cells. Follicular cells, and the whole structure is known as a follicle. So the primary oocytes are present inside a structure called the primordial follicle. Now let's demagnify it and get back to our original slide. So that is the place where we have our primary oocytes. Now what happens to primary oocytes? These primary oocytes undergo mitotic division and they do not complete it. Now these primary oocytes just start to undergo meiotic division, which is a reductional division. What happens in reductional division is that a cell which is diploid and has double the amount of DNA. See, when we write double the amount of DNA, we use the notation 4C. What does this mean? This means it has 4 chromosomes for one thing. See, we inherit 1 chromosome from our father, 1 from our mother. And if we double up, how many chromosomes do we get? We get 4, and that is what 4C means. It simply means twice the amount of DNA. So now the primary oocyte is in this particular condition. If it has to undergo meiotic division, it goes through 2 phases, meiosis 1 and meiosis 2. We are not going to get into the details of it, but what we need to know at the moment is that the primary oocytes, when they undergo start to undergo meiosis, they simply get arrested at a stage called prophase 1. I want you to recall the characteristics of prophase 1. During prophase 1, there is recombination or exchange of genes between homologous chromosomes. So just at this particular spot, the primary oocytes are arrested. They do not proceed with meiosis any further. And where is all of this process happening? This is happening in the embryo. And now our primary oocytes are somewhere here in the primary follicles. Now we will not inside the primordial follicles. Now we will not look at primary follicles, secondary follicles, and graphene follicles right away. We'll look at them in our future lectures, but let us see what happens next. Now once puberty is hit, once now the embryo will grow up, and then we will have a healthy girl child. Girl child will be born, and then the girl child will progress through her life. She'll pass through the teenage, 17, 18, 19 years, and then she'll hit her puberty. Puberty has a variable time period. From 16 to 19 years, puberty can be hit at any point of time, right? So when puberty is hit, there are certain hormonal changes in the body, and one very important hormonal change that is important for us to know with respect to oogenesis is the change in the hormonal secretions from the brain. Now in the brain, in the human brain, let me just draw a rough sketch of the human brain. In the human brain, we have got a structure called the hypothalamus, and we also have a structure called the pituitary. So let us say this is the hypothalamus, and this is the structure called the pituitary. Now I'll magnify the pituitary. Pituitary has two lobes, so it appears somewhat like this. In the pituitary, we have got something called as the anterior lobe, and then we have got another lobe called the posterior lobe. I will just come to this. Now when puberty is hit, the structure called hypothalamus in the brain starts to secrete considerable amounts of hormone called GnRH, and this stands for gonadotrophin releasing hormone, a hormone that will release gonadotrophins. That is what this name means, gonadotrophin releasing hormones. Now let's use this yellow. So these hormones are released, and where are the target cells for these hormones? The anterior pituitary. So these hormones, they reach the anterior pituitary here, and here this particular hormone, gonadotrophin releasing hormone, stimulates the anterior lobe of the pituitary. And what the anterior lobe of pituitary does, it starts to make two hormones. It makes two hormones. The first hormone is called the luteinizing hormone, and the other hormone is called follicle stimulating hormone. The luteinizing hormone, let me write it down, luteinizing hormone, and the other hormone is called follicle stimulating hormone. So we got two hormones here that are secreted by the anterior lobe. Now these hormones post puberty, they are secreted into the blood because these are endocrine glands. Pituitary gland is also an endocrine gland. An endocrine gland will release its secretions, it's produced into the bloodstream. Now the bloodstream will take these hormones to all the parts of the body, but the target cells of these hormones, they lie in the ovary. And where are those target cells? The target cells are these follicles. I hope you remember when I just drew the follicular cells for you, the granulosa cells. Here we have the follicular cells and the granulosa cells. These are the targets for the luteinizing hormone and the follicle stimulating hormone. Now, upon reception of these hormones, the primary oocyte, which was arrested in prophase 1. You remember? Getting back. So, this primary oocyte had started to undergo meiosis, but the moment it starts meiosis, it reaches prophase 1, which is the first stage of meiosis 1, where recombination was taking place. The cell is arrested. All the metabolic activities are arrested. But now, exactly at the point of puberty, these hormones tell this particular primary oocyte to proceed further. Now, this particular primary oocyte undergoes metaphase 1, and anaphase 1, and telophase 1. And meiosis 1 is completed, and at the end of first meiotic events, two cells are generated. Now the first cell, usually both the cells have the same size, but in this case, in case of oogenesis, only one cell is extremely large because it gets the lion's share of the cytoplasm and all the organelles in it. So one cell becomes extremely large and the other cell is extremely small, very tiny. It has barely any, very little meager amount of cytoplasm and organelles, if any. Now this large cell here, which is formed by the first meiotic division event of primary oocyte, see it divides, this cell is known as the secondary oocyte here. So we have the secondary oocyte here. But now, and let's just wait here. And the other structure that is formed is known as the first polar body. Now the first polar body is extremely small. The secondary oocyte is pretty large. And this is the end of meiosis 1. Now this secondary oocyte, now let me tell you in the diagram where has this taken place. So let's go back. So we had primordial follicles which had the primary oocyte, and here meiosis had just frozen. The cells were frozen in prophase 1, and the child was born with this particular ovary with millions of primordial follicles. Remember, there are no primary follicles in the embryo, no secondary follicle, no Graafian follicles, and these 4 follicles are important for you to know. Now here we have the primary oocytes. Now, post puberty, these primary oocytes receive the hormones, the thing that we just discussed, and then more granular cells are added to them and they turn into something called as the primary follicles. And these primary follicles then receive more of the hormones, they get converted into the secondary follicle. Now, here in the primary follicle, these are those follicles, and let me just magnify. So, here we have a primary follicle. Now you see, let me show you primordial follicle. See, a single layer of granulosa cells, and here I've got primary follicles with so many layers of cells, large number of granulosa cells. And in the center, if you see carefully here, this is our secondary oocyte with the nucleus. So this is the secondary oocyte, and polar body is lying somewhere around. So let me just draw a tiny little dot, and this is the first polar body, and this is the secondary oocyte. So if you're asked where is the secondary oocyte found, it is in the primary follicles. And then the same secondary oocyte and the polar body are then also transferred to the secondary follicle. They are not transferred, actually the whole primary follicle gets transformed into the secondary follicle. We'll see to that when we'll come to ovulation in the future classes. Now let us just go back to the remaining events. Let's demagnify it slightly more so that we can accommodate everything. Now we were here. So primary oocyte has been divided. We get a secondary oocyte, a first polar body. Another important thing that you must remember is the secondary oocyte. See, the content, the amount of DNA was 4C. Even here, primary oocyte also had the same 4C because the oogonia underwent mitotic events. So the ploidy is 2N, this is 4C. So let us write 2N and 4C even for the primary oocyte. So this is also the condition for the primary oocyte. When primary oocyte divides into 2, its DNA is halved. So here the amount of DNA is 2C, and here also the amount of DNA is 2C. But what is the ploidy? The ploidy is no more diploid. The cell is no more diploid. The cells are haploid. And I hope you've learned this in meiosis, but anyways, I am also going to provide you with some supplementary material where you can again recall why the products of meiosis 1 are haploid. But at the moment, you must remember that at the end of meiosis, we get two cells, the first polar body, the secondary oocyte. Both of them are haploid, but they still have double the amount of DNA. Right, now we'll go to the next part. Now what happens that the secondary oocyte, exactly now, the secondary oocyte and the granular cells are increasing, growing around the follicles, and the follicle is at the center. These cells are around these are called the granulosa cells. Anyways, let's not focus much there, and let's look at the secondary oocyte here. Now the secondary oocyte also starts to undergo the second meiotic phase, and let us try to understand it on a new page. Let me get a new page, and here I have a new page, but this seems to be messy, so let me just add up a blank page here. So here I have a new page. Now let us just draw. So we had this one cell, and this gave rise to the oocyte and the first polar body, and this is meiosis 1, and this was, so we have the primary oocyte and here, and now we have the secondary oocyte and the first polar body. And the condition here of both of these cells is both of them are haploid and they still have double the amount of DNA, or the same amount of DNA that a normal diploid cell has. Anyways, but the ploidy has been changed. Now both the secondary oocyte and the polar body have to undergo meiosis 2. So again there are 4 stages. We got the prophase 2, metaphase 2, anaphase 2 and telophase 2. You must bear in mind that prophase 2 is a rare event. It usually does not take place in a lot of cells, but it does take place in a couple of cells. So anyways, so we still have all of these phases that the cell must undergo. Both the polar body will also undergo these 4 stages, the second meiotic event. So now the moment these particular events begin in both of these cells, this secondary oocyte has to and these changes are going to take place within the nucleus. So before cytokinesis, before we get 2 cells from here, we will have events taking place inside the nucleus. So before cytokinesis, karyokinesis takes place, division of the nucleus. So here the events start to take place. Prophase 1 and metaphase 1. The chromosomes that were here will be aligned on a metaphase plate like this, right? So what happens? The cell, secondary oocyte, starts to undergo meiosis 2. Polar body also starts to undergo meiosis 2. But both of these structures, again, for the second time, are now frozen at metaphase 2 of meiosis 2. So again, they are once again frozen. And where are these cells? So see, let's look at this. Completion of meiosis and onset of meiosis 2. So when meiosis 2 starts, secondary oocyte, which was formed here, it starts to undergo meiosis 2, but then it is arrested at metaphase of meiosis 2. So it cannot proceed any further than meiosis 2, and same is the fate of polar body. One important interesting thing to remember is that polar body may or may not undergo meiosis 2. We'll see to that why that happens, or what is there any significance of it. Anyway, so where is the secondary oocyte here in the diagram? So the secondary oocyte is usually formed in the secondary follicle. It is usually formed here. So if you look at the center, let us magnify. So the secondary oocyte is here. This is the secondary oocyte now. So we have the secondary oocyte, and we have the just give me a moment and let's just go back and see. So we got the secondary oocyte and the first polar body. I think I've made a tiny little mistake before. So we got the primary oocyte and we got a first polar body here, and I've made a tiny little mistake before this, and let me correct it. In the primary follicles, right, in the primary follicles, we have the primary oocyte, which has just started to undergo meiosis 1. So there would be no polar body here. There wouldn't be any polar body that I mistakenly have drawn here. So there'll be no polar body here, but we'll have a polar body at the end of the secondary oocyte formation, which forms in the secondary follicle. So we have the, I hope this content is clear, and the confusion that I just created has also been sorted out. So let me just straighten up the image and then we'll proceed to the next part. So now we have the secondary oocyte, but now you must be wondering, when will the egg be formed? When will be the male gamete formed? So the male gamete, the female gamete, when will it be formed? So the female gamete doesn't form so easily. I hope I have not lost my sound here. Once again, just like the last time. So now the secondary oocyte, which is in the secondary follicle. Now what happens? In this secondary follicle, more cells are added up and a new structure called the graphene follicle is formed, and the graphene follicle eventually moves closer to the surface wall of the ovary, and the surface wall of the ovary then ruptures and it releases this secondary oocyte, right? So can you see, this is released, and this process of release of the egg from the ovary is called ovulation, and we'll get to ovulation in our next lecture. Ovulation. Now what is released in the ovulation? Let us magnify, get closer here, and during ovulation what has been released is we have a secondary oocyte. This is a secondary oocyte. It is haploid, but it still has double the amount of DNA, and we also have a polar body somewhere around, and the polar body is also haploid, but it still has double the amount of DNA. Now it is still not the female gamete. The female gamete should be haploid, and it should only have one set of chromosome, not a pair of chromosome. We have two chromosomes here still, so we need only a single set of chromosome here, right? Now when will that be formed? When will the female gamete be formed? And the female gamete will be formed in just a little while. So what is the fate of the secondary oocyte? Once this this secondary oocyte with all of these cells around it, this particular structure can also be, you can also call it the immature ova, or the female gamete, immature female gamete. Now once this female immature ova is released, there are structures, and let's go to the next slide so that I can get a little bit of space. Let me rub all of this up and make some little space for myself. Now let me draw this ovary. So let us say we've got this ovary here, and this ovary has released this particular structure, which is that immature ovum or immature egg. Now there are these structures which are known as the fimbriae, finger-like projections, and they are a part, uh, and they are a part of the fallopian tube. So we see we have a fallopian tube here, or we also call it the oviduct, and this structure here, these are called fimbriae, and the fimbriae will absorb or take in the immature ova or the egg inside it. So once the egg is inside, what happens next is what we'll see here. Now let me just magnify on this particular structure here and just bring it to the center. Right now here what we have is, so you see the secondary oocyte, which is haploid but has still double the amount of chromosomes, double the amount of DNA compared to a normal gamete, right? So this is taken up by these fimbriae, the finger-like projections, and then this enters the fallopian tube. Now if there are male gametes, the sperms, if the sperms are already inside the fallopian tube, if the act of copulation or coitus has taken place, if the male and female have exchanged their, if the males have deposited their gametes into the female reproductive system, then the sperms are traveling and they are inside the fallopian tube. And in the fallopian tube, what happens? Let me draw this. So let's say this is the structure, this is the immature egg with a large number of granular cells around, and inside we've got the secondary oocyte, and here we've got the polar body. So there is a layer of cell, and let me just draw it with red. There is a layer of cell around it, and let me just now give it certain names. So here, these outer layer of cells, these granular cells, these ones that you see here, dotted structures, these are called corona radiata, which means the radiations from the sun. Corona is sun, radiata is radiations, because they appear like that. Not in my drawing, but here at least you can appreciate it. And then this red line that I have drawn is a layer of protein which is called zona pellucida. And inside that, now here we have one cell, the large cell, which is the secondary oocyte. And here we've got another cell which is called the polar body. And which one? The first polar body. Now the male gametes, let me draw them, and there are a large number of male gametes. If the male gametes are in the fallopian tube and one of the male gametes happen to come in contact with zona pellucida, it penetrates these corona radiata cells and comes in contact with zona pellucida. Once this happens, once this event of fusion of the male gamete or collision of the male gamete with zona pellucida happens, the secondary oocyte undergoes meiosis II. Recall that secondary oocyte is arrested at metaphase 2 of meiosis II. So the moment this collision happens, the secondary oocyte proceeds beyond metaphase 2. It goes through anaphase 2, telophase 2, and then it divides into two cells. So we get two cells. One cell is large, but then secondary oocyte forms again a very tiny structure, right? So the secondary oocyte now has formed a large cell with most of the cytoplasm just like it had and all the organelles that it had, and it forms another small cell just like the first polar body, and this smaller cell is called the secondary polar body. And this structure here, the secondary oocyte that has now divided, is the egg or the ova, the mature egg or the mature ova. And once this is formed, the nucleus that is present inside the sperm goes and fuses with it, and then we have the fertilization and we get an embryo, right? We get a zygote to start with. We directly don't get the embryo, we get the zygote to start with. We get the zygote, and then the zygote multiplies through various stages and forms the embryo. Now also, very interestingly, you remember this first polar body? Sometimes, it's very rare, but sometimes this first polar body also undergoes division. Because it is so small, it forms two very small polar bodies. And so in totality, at the end of oogenesis, and oogenesis can only be completed if the sperm fertilizes the secondary oocyte. So at the completion of oogenesis, we get three polar bodies and we get one egg, and the egg actually will then fuse, form the zygote and the embryo. The three polar bodies are eventually discarded. They are of no use. Now you must ask, you might think, what is the point in making three polar bodies? The point in making three polar bodies is to get rid of that excess amount of DNA. You remember, we started off with let us go back and look at the diagram. We started off with the primary oocyte, which was diploid and had double the amount of DNA. Let's look here. We started off with this. What was the condition? It was diploid and we had 4 chromosomes. At the end of meiosis 1, we were successful in making haploid cell, but at the end of meiosis 1 here, we still had double the amount of DNA. Let's go back down now. Okay, and here. So now these polar bodies, each of them have only one set of chromosomes. So you see, not only the polar bodies help the egg to become haploid, but also it helps the egg to have only one set of chromosomes. And this is all about oogenesis. Thank you.